Hi, my name is Lucy Pavlovich, and I would be talking about string theory, but I think that's too much to talk about in a 10-minute video. So instead, I'm going to talk about robots. Well, not so much killer robots that are going to take over the world like you see in sci-fi books and movies, but rather a bioengineering innovation. As of now, there is a robotic arm that can be controlled by thought using a chip implanted into the brain. There is a way that a robotic arm can be attached to a patient without having to go through the brain. The problem is that this would only work if you could do the procedure right when the person's limb is amputated, because it requires reconnecting their nerve endings to other tissues in the body and connecting chips there. However, this isn't possible much of the time because people will need emergency amputations, or if they get caught in a bomb explosion, that isn't exactly the pristine lab procedure to ensure this reconnection. So, I developed a theoretical process in which a chip in a newly generated muscle in the arm allows you to move a robotic arm. This theoretically would work for anyone who has had an amputation, either recently or years ago. How, you may ask? Well, let me explain. When a person has a limb amputated, the motor neurons and sensory neurons are cut in half and begin to degenerate because there is no sensational feedback. We are not lizards. We cannot just grow back limbs. And if we tried, we all know what would happen. Spider-Man. So instead, one concept is to take people's skin cells and revert them back to cells similar to stem cells using a process of IPS, induced pluripotent stem cells. 2012 Nobel Prize winner Shinya Yamanaka developed this process. This could be revolutionary to the field of regenerative medicine because they can divide into any cell needed, such as neurons, heart, pancreatic, and liver cells. These IPS cells act like embryonic cells. They replicate and can be told to form into any certain type of cell based on the environment you put them in. In theory, once the skin cells are reverted back to stem cells, they can be separated and cultured into two groups, motor neurons and muscle tissue, by using specific chemicals. The motor neurons will be told to grow into only one specific set of neurons, and the muscle tissue will be cultured around electrical impulse sensors. This allows the neuron to communicate with the robotic arm. Electrical impulse sensors work by measuring voltage fluctuation resulting from the ionic current within neurons. The electrical impulse that is sensed is the basic function of a neuron. Neurons are actually a fascinating thing. They are the one cell that allows electricity to run through them. They do this by polarizing and depolarizing every 10,000th of a second. Think about it. Right now I am talking. Every time my mouth and tongue move, motor neurons are firing and shocking my muscles into doing what I tell them to do. How do they do this? Well, a message is sent via neurotransmitter, chemicals, to tell the neuron to start firing. The dendrite, the receiving end of the neuron, has receptors to catch these neurotransmitters so that they know what to do. Then they start firing. Electricity flows down the axon, being propelled by the imbalance of ions on the outside and the inside of the axon. Once it has reached the end, the electrical impulse tells the end of the axon to release neurotransmitters, and the process starts over again with the next neuron. The electrical impulse is recorded and interpreted into different movements that are then reenacted by the robotic arm. After the motor neurons are cultured, an amputee would get a stem cell injection allowing them to regrow the motor neuron. Along with that, the new muscle tissue that has been cultured around the sensor will be added to the end of the person's amputation, and the neuron will grow to connect itself to that. Hopefully, this process will connect the spinal cord to the new muscle tissue. Then the person's body should be able to send signals through the neuron that the sensors will be able to pick up. Programming deals with the rest. The sensor readings are analyzed and converted into a movement in the robotic arm depending on where the electrical impulse was, which sensor was detected, and the amount of electricity sent and the time lapse between impulses. If, for example, sensor 1 detects a signal, then the thumb will move. The robotic arm will know how much to move and how quickly to move the thumb based on the amount of electricity and the time lapse between shocks. This could be a revolutionary idea that I find very interesting, and I hope this video has helped to explain this concept, or even better, has gotten you interested in the field of bioengineering. Thanks for watching. Wow, this was a short video. Maybe I could have talked about string theory.